Now that we've talked about amino acids, let's talk about peptides, because this is what you assemble amino acids into to make proteins. So a peptide, by definition, is just two or more amino acids that are joined by a peptide bond. And peptide bonds are formed through a condensation reaction, so that's where a molecule of water is lost. So up here we have two amino acids, and one of them, this is the carboxylic acid group, and then one, here's the amino group. And so in the reaction that forms the peptide bond, this hydrogen and this OH are lost to form water, and then you form a bond between this carbon and this nitrogen. So between these two here. So this is what you get down here. This right here is the peptide bond, and it has this characteristic arrangement where you have a one carbon atom double bonded to an oxygen, and then the nitrogen that is bound to has a hydrogen attached to it, and then each of those attaches back to the alpha carbon of the adjacent amino acid. So this is the alpha carbon of amino acid 1, and this is the alpha carbon for amino acid 2. And here, you notice we put just R1, R2 for the different residues because we don't care, or the different side chains because we don't care what they are right now. The point is now we've connected these two together in a bond. And of course, breaking a peptide bond requires water, so that's a hydrolysis reaction. Now, of course, these don't happen spontaneously. Peptide bonds, for the most part, are produced when making proteins by ribosomes. So ribosomes have enzymatic activity that help produce peptide bonds, and then there are other enzymes that help break peptide bonds. So they go by lots of different names. Peptidases are collectively what they're known as, and their job again is to catalyze the breakage of this bond using water. And then once you have two amino acids connected together, you have a peptide. Now, if you only have two, it's a dipeptide. So this, for example, would be a pentapeptide because there are 5 amino acids all connected together with 4 peptide bonds connecting them. So the amino acids in this situation are serine. So this is serine. This is serine. And then this is a glycine. This is a tyrosine. This is an alanine, and this is leucine. So the full long name of this whole molecule now would be seroglycyl tyrosyl alanyl leucine. But of course, we don't usually refer to them that way. Most peptides that have any biological function have some special name, usually just the name of a protein. But this is the formal name for this particular pentapeptide. And then sometimes you'll see them an amino acid sequence written out with the 3-letter abbreviation. So this is the full long name. This is the same thing, just with the single letter abbreviations for those amino acids. Now, one other thing to say about the notation here: the name, or the way we list off the amino acids in a peptide, is always starting from the end of the peptide that has a free amino terminal. So we call this over here the amino terminal because it has a free amino end. It's not attached to anything. Sometimes it's just called the N terminal. And then down here you have the carboxyl terminal, or carboxy terminal sometimes. And sometimes it's also just referred to as the C terminal. By convention, whenever we write out the amino acids in a peptide, we start with the amino terminal first and then end with the carboxy terminal. So in all 3 cases here, we have serine listed first to the left, then glycine, tyrosine, alanine, leucine, and leucine is last because it is the carboxy terminal. Now, sometimes you will see that written out explicitly. So you might see an amino acid sequence where an actual little N or a C is here to indicate which end is the amino terminal and which end is the carboxy terminal. But oftentimes it'll just be, they won't be there at all. And so it's implied that you have the N terminal at one end at the left and the C terminal to the right. And then these are, again, peptides come in lots of shapes and sizes. So by definition, a peptide, the broadest definition is just any number of amino acids that are linked together by peptide bonds. But in practice, usually when you hear the word peptide, it's used to refer to very short amino acid or very short sequences of amino acids. So for example, this molecule here is called oxytocin, and it only has 9 amino acids. So this would be a short peptide, whereas this molecule over here, it's part of a very large protein called titin. It's actually part of the largest protein that has ever been discovered, and it has 420 amino acids. But again, you wouldn't probably hear it referred to as a peptide, whereas something like oxytocin is sometimes referred to as a peptide. And there's not really an objective line to draw where you would say something is long enough to be a peptide versus a protein. So sometimes those terms are used interchangeably. But a lot of peptides, like oxytocin, are important as signaling molecules. So a lot of hormones, for example, are short peptides like this, and other things that might be secreted out of a cell. And so that's where those terms will show up. Also, molecules like oxytocin, these short peptides, often start off as longer peptides. So they're actually translated like other proteins, but actually as a longer sequence of amino acids, but parts of it get cut into a smaller piece. So that we'll talk more about how proteins are actually made and processed later on. And there are then, of course, lots of things you can do with proteins. A lot of proteins, perhaps most proteins, in fact, eventually get attached to other types of molecules. So when something is attached to a protein, we call it a conjugated protein. So something has been conjugated to it. And the thing that gets conjugated to a protein we call a prosthetic group. So you can attach lots of different types of molecules to proteins. So a lipid, for example, attached to a protein will give you a lipoprotein. A carbohydrate attached to a protein is called a glycoprotein. Phosphate groups are really important. Phosphorylated proteins behave much differently than their non-phosphorylated forms. So adding and taking away phosphates is one way to change the function of a protein. Big molecules like hemes and flavins are also important. They are important parts of enzymes, or in the case of heme, for carrying around things like oxygen. And then you can attach metals to proteins too. So lots of proteins have metals conjugated to them. Iron, for example, is conjugated to hemoglobin through that heme group. And then there are lots of other metals out there that are important. So those are called metalloproteins. So a protein with a metal conjugated to it is a metalloprotein. And of course, these are importantly conjugated after translation. So in other words, these groups are added to the protein to specific amino acid residues after the protein has been made. And we're going to come back and talk about protein structure a lot more in later chapters. So we will not go into more detail right now. But the other thing we should talk about in relation to proteins is how you work with and study proteins. So proteins, of course, are big molecules for the most part. And if you extract cells or tissue from an organism, and then even if you grind it up, it's going to be a mixture of a bunch of different types of molecules. So there will be lipids that are from the cell membrane, there will be carbohydrates, there will be some nucleic acids in there, and there will be proteins, and there will be lots of different types of proteins. So if you are interested, if you want to find just a particular protein in a tissue cell sample or a cell sample, you need to first isolate the proteins from the other cellular components, and then second, isolate the protein you want from the mixture of proteins. And there's lots of ways to do this, and I'm just going to give you some examples. Usually the first thing you want to do is get the non-protein bits separated from the protein bits, and there are, again, lots of ways to do this. Usually the first thing you do is you grind up tissue that you've collected, and that smashes the cells so that the cell contents are separated or escape from the cell membrane. And then usually through centrifugation, you then pull all those large cellular components out from the smaller molecules that are just floating around in the cytosol. And then you have a solution that, again, just contains proteins plus other molecules. And then you have to have, there are various ways to then purify or isolate the proteins from the non-protein parts of the cell. One is using salt precipitation. So proteins have the tendency to precipitate out of solution in the presence of high salt concentration. So one common way to purify proteins from a mixture is to add high concentrations of ammonium sulfate. That causes the protein to precipitate into a solid, which again you can then centrifuge, remove the liquid part, which again would contain all your other non-protein constituents, and then you just are left with the protein part. Now, of course, once you've added, or once you've used salt to precipitate proteins, you need to remove the salt usually, and then other kind of small molecules that might have precipitated out with it. And there are different ways to do this. One common technique is called dialysis. So dialysis uses the principle of osmosis or diffusion to get rid of salt and to concentrate down, sometimes to concentrate the protein more. So if you put your salt solution or your protein precipitate with the salt into a container of water, which in turn is in a dialysis bag. So the dialysis bag is this little structure here, and it's basically just a thin membrane that is porous enough that it allows small molecules like salts to go in and out, but not so much that the large protein molecules can escape. So if you put that into a beaker of pure water, then the salts will diffuse out and the proteins will be left behind. And you can, there's other forms of dialysis where you can put actually your protein sample into a high salt solution. And a high salt solution, and then in a membrane that's only permeable to water, and then water will actually enter, water will leave the dialysis bag through osmosis, and you can actually then raise the concentration of your protein by getting rid of that water. But in any case, what you've done now is now you've got a solution which contains hopefully just a mixture of proteins and a smaller concentration of salt. So now you have all the proteins separated from all the other cellular components, but again, usually what you want to know is, or you want to be able to study just one protein out of that mixture. But again, in any given cell, there are going to be hundreds or thousands of different types of proteins. So you have to figure out a way to separate that protein from the other proteins. So there are lots of ways to do this, but they all basically involve using different physical and chemical properties of different types of proteins. So like I said, proteins come in all shapes and sizes, and we'll talk about how protein shape affects their function later on. But differences in size, molecular weight, differences in electrical charge, and other properties you can exploit to separate those proteins from their other cellular neighbors. One common way to separate proteins from each other, and in fact other types of molecules as well, so not just proteins, is called column chromatography. So in column chromatography, you make a column, which is just a big long tube. So this is your column here, and it's just a big glass or plastic tube filled with some sort of resin, what's called the stationary phase, and usually it's little beads of silica. There are other things you can use in columns, and then it's usually in typical setups, the column is set up vertically so that the fluid that contains your protein. So the idea is that the protein you want is somewhere in this reservoir, but again, mixed with a whole bunch of other proteins that maybe you don't want. So then you either pump the fluid out of this reservoir or just pour it onto the top of this column, which again is just a big tube. The fluid will flow through the column and then come out the other end, because at the other end there's a little tiny hole with some tubing that will let the liquid go through, but will not let the solid phase go through. That's why it's called the solid or stationary phase. So the fluid goes through, and some property of the resin will separate the proteins by letting some proteins go faster than others. So proteins that go more quickly past the stationary phase will come out of the tube first, whereas proteins that come out or get stuck for whatever reason on the stationary phase will take longer to come out, and so they will come out last. So then at the bottom of your column, you put a series of tubes that will collect the liquid that comes out, which we call fraction. So sometimes this is called a fraction collector. If for fancier labs, you might have a little machine that contains tubes on like a rotor, and it turns around at a certain rate to collect specific volumes. But the old-fashioned way is just to put the tubes in a rack and just move them from one spot to another after a certain amount of time, or after a certain amount of volume has come out. And then hopefully what happens is whatever comes out contains your different proteins, and then usually it's attached to some sort of detector. So there are lots of ways to detect proteins in a solution, and so you can use that property to measure how much of whatever it is. It may be other molecules too, but whatever comes out, you can see which tubes contain the highest concentration of different substances. And then the idea is that one of those fractions, or one or two of them, will contain just the protein you want and not the proteins you don't want. So how does it actually separate the proteins? There's different ways to do this. So one method is called ion exchange chromatography. Here you have a resin which contains negatively charged functional groups. So some covalently bound to these resin molecules, there is a substance that keeps a negative charge at whatever pH you're using. And then remember that proteins, just like individual amino acids, can be charged if they have charged side chains. And then the amount of charge depends on the number of side chains, and it depends on the pH. So if you know what the charge properties of your protein are, you can choose a pH that will make the protein either positive or negative, and then let the protein mixture go through. If your protein, the protein you want, has a large net positive charge, then it will be attracted to the negatively charged resin beads, and so it will go slower, whereas negatively charged proteins will go through faster, and they will come out in your first or your early fractions. And of course, so it depends on which one you want. If you want the negatively charged proteins, you would collect the early fractions. If you wanted the positively charged proteins, you would collect the later fractions. And then neutral proteins would come out somewhere in the middle. And then you can also use chromatography to separate proteins or other molecules by size. So this takes these beads that have these porous holes in them. So these beads are made of a polymer, and they're not solid. They have little tiny holes, and the holes are small enough that small proteins or molecules below a certain size will kind of get stuck and will have to take a longer path essentially to get from the top of the column to the bottom. Whereas bigger molecules don't get stuck in those little holes. They just, because they don't fit, they just go around the beads, and they end up coming out faster. So this is one way you can separate by size. Now, this is not necessarily separating by molecular weight, because the shape of the protein also matters. So you could have two proteins that are the same molecular weight or very similar molecular weight, but one might go through this kind of column more quickly than the other if the shape is different. So a spherical or roughly spherical protein that's small would probably go slower through a column like this than a protein that's about the same molecular weight but maybe is sort of elongated or fibrous in shape, and so that matters too. So this is not a way to separate necessarily by molecular weight, but it does help let you separate by size. And then if your protein binds to a specific molecule and you know what that molecule is, then you can use that as a sort of a trap, a way to grab the protein you want as it goes through the column. This is called affinity chromatography. So proteins, and we'll learn about this more later, but most proteins work by binding to something else, either other proteins or other small molecules. And so they have binding sites for those substances. Sometimes if it's a small molecule, we call that substance a ligand. And so if you take that ligand and covalently attach it to these resin beads in your column, then you can pass your protein mixture over the column. Hopefully all the proteins that don't bind to your ligand will just go right through, whereas the protein that you want, the protein of interest, which hopefully has a high affinity for that ligand, will get stuck. And then you'll have gotten the unwanted proteins out, washed through, but then you have the problem that now you need to get the protein you do want off of the column, because especially if there's a high affinity between the ligand and the protein will just sit in that column forever, no matter how much fluid you pass over it. So to get the protein of interest off, you pass another solution over the column. This one contains the ligand. So now the ligand is not dissolved, or it is dissolved in a solution. It's not physically attached to these resin beads. So as this fluid passes over, the idea is that the ligand is more concentrated in this solution than it is on the beads or on the column. So the protein will bind to the free flowing ligand, and then both will come out at the other end of your fraction collector. And then later on, if you need to, you can use other techniques to separate the ligand and the protein. These are non-covalent interactions. So usually when a protein binds to its ligand, it's doing so just using either ionic interactions or hydrophobic interactions, or hydrogen bonding. So usually it's not that difficult to separate them once they're off of your column. So these are all techniques that you can use to purify a protein from a mixture of other types of proteins. But once you've purified your protein, you may want to identify it, or at least make sure you got the protein you wanted. And there are lots of ways to identify a protein in a mixture or in a purified sample. A really common and important type of protein identification involves this technique called gel electrophoresis. So gel electrophoresis takes advantage of the fact that proteins have a charge to them usually. And so if you put proteins in an electric field, they will move just like any other charged particle. So in a gel electrophoresis, you make a gel. So the gel can be made of different materials. This is down here, the gel, this sort of blue thing. And it can be made of different materials. For protein, typically the gels are made of this stuff called polyacrylamide. So sometimes this process is called polyacrylamide gel electrophoresis, or just PAGE, P-A-G-E. And so this gel is made of polyacrylamide, and at one end, so at the top of the gel, there are these little fluid-filled wells. They're just empty holes that are filled with usually some kind of buffer. And you take a pipette and you add small quantities of your protein mixture or other samples to these wells. And then the chamber that the gel is in, again, it's filled with buffer, and there are these electrodes in the solution. One's negative, one's positive. So you hook that up to a voltage, and current will pass through the gel. Actually, strictly speaking, the current will move this way, the current will move up. But the proteins, if they're negatively charged, will move down. Now, there's different ways to do this. In fact, you don't have to have the wells at the top. You could have wells in the middle. And so, in fact, if you had a mixture of negatively and positively charged amino acids, the negatively charged amino acids would go down, positively charged amino acids would go up. But again, this technique works better if you separate by size. So again, you want to separate by size, not by charge. So to deal with that problem, to deal with the fact that different proteins can have different charges depending on pH and everything else, you use this stuff here called SDS. SDS stands for sodium dodecyl sulfate. Basically, it's a type of detergent, and detergents just stick to things. That's how they work, and they make things more soluble by sticking to pieces of dirt or oil. And so that's what SDS does to proteins. It sticks to the proteins, and what it does is each SDS molecule adds a negative charge to the protein, because SDS molecules are negatively charged, and so you add enough SDS that the protein is completely surrounded by SDS molecules, and so it's effectively completely negatively charged regardless of the number of charged amino acids. And the number of SDS molecules is going to be a function of the size of the protein. So that means the total negative charge of the protein will be a function of its size. And what that means is that the protein will move through the gel at a speed that is proportional to its size. In fact, to its molecular weight, because SDS is also what we call a denaturing agent, and you add other denaturing agents, which means that it causes the proteins to unfold. So it means the shape doesn't matter. It doesn't matter if they're cylindrical or spherical or what. Now there are what we call native gels, where you might want to run the gel through, or the proteins through the gel without changing their shape, or with them being in their native shape. But if you're just interested in molecular weight, you can do the SDS gel, which is only separates by size. And then also, of course, with your gel or with your sample, you load some sort of dye. Proteins themselves are invisible most of the time, especially at the concentrations you would use in one of these experiments. So you add a dye that also is charged and that will also move through the gel, and that will be visible. So you can see it moving through the gel, and you can then see how far your sample has gone. Hopefully your dye is smaller than any of your proteins, because it will move the fastest, and you don't let your protein run off the end, which can happen. And then after the gel runs, usually you add some sort of another dye, some sort of pigment that will stick to the proteins, and then will show you where the proteins ended up. So over here, this is an example of an electrophoresis gel. So each of these purple bands represents a protein or a group of proteins that migrated to that distance. So the top of the gel, so somewhere up here, presumably, would be where the wells were. And so into each of those wells, what they did for this gel is they added some sort of, or some mixture of proteins, and then they put them in, mix them with SDS, and let them run. Typically, when you do an SDS gel, you have a marker lane, because this will be a mixture of proteins that you already know the weight of. So you can look and see, well, I know that there are 1, 2, 3, 4, 5, 6 bands, or 5, 6 different proteins in my marker mixture. I know how big they are. So for example, I know that this band corresponds to a protein that has a molecular weight of 66,200. And so you can use that to figure out the molecular weight of the protein. So we'll talk about that in a second. And so what this shows is that they put into each well here a different mixture of proteins that had different things done to them. So it looks like what they did here, I have no idea where, I think your book probably describes in more detail what happened here. But this lane here contained protein extract from a cell culture that looked like, it says it's uninduced, which means probably that's their control. So they didn't do anything special with those cells. And then this next column contains extract from the induced cells, which means that they did something to those cells to make them express a particular protein. And so what you can see is the main difference is there's this large band here, which means that inducing the cells, whatever that means, caused this protein to show up, which was not present in the uninduced cells. And then they go through different extraction methods to purify that protein. So here this is the soluble crude extract. So that means this whole column just contains all the proteins that come out when you extract protein from that cell. And then this is the column after they did the ammonium sulfate precipitation. So presumably this protein precipitates really well from ammonium sulfate, and that purified it further. And then they did actually anion and cation exchange. So that means that they did ion exchange with negatively charged ions and they also did negatively charged beads and with positively charged beads. So that again just purifies it even more. And then over here, presumably is another their other control. So this is the protein that they were looking for, already purified for them, and so they can tell that it was there as well. So that's what that band represents. That's the protein they were trying to target, and the gel separated it from its neighbors. But like I said, and so you can use gel electrophoresis. Hypothetically, you can use it to purify proteins, but electrophoresis is actually a bad way to purify proteins. First, because it only works on a small volume. Second, because the protein is then stuck in this gel, and to get it out is kind of a big pain. You have to like cut it out with a razor blade and then boil the gel, and it's a whole hassle. So that's why chromatography and those other techniques are better for purification, but gels are great for identification. So what you can do, for example, is you can measure the molecular weight of your protein. So if you don't know the molecular weight of your protein, or if you want to find a protein that has a particular molecular weight, you can use gel electrophoresis. So like I said, this is pretty typical. You would always have one column that's your standard. So here we've got a bunch of proteins that we know what their molecular weight is. And over here in this column, this contains our unknown protein, which means we don't know how big it is, or we don't know exactly where it's going to go on the gel. So here we can see that this unknown protein ends up right about at this level on the gel. So right about there, somewhere between 45,000 and 21,000 molecular weight, or 45 and 31, I'm sorry. So to get a more precise measurement of the molecular weight, you can then graph the relative migration of each of those standards to its molecular weight. So again, you measure usually the distance from the well. Actually, it's the other way. You look for the dye front. So if the dye front is down here, you say, okay, the dye front migrated this far, right? And then this band here, it migrated this distance. So this distance from the well to this point, and that distance divided by the total distance of the dye front is its relative migration. So you graph that out. So for 14.4 thousand Daltons, you say, okay, well, that's probably going to be this point here, and so that's going to be whatever that distance is, and then this the y-axis here, the point on the y-axis will be again the molecular weight of that protein. So 14,400. And you do that for every one of your standard proteins. And then you see how far did my unknown protein go. So again, you measure this distance. How far is it from there to the well? And then how far is it from there to, again, my dye front, presumably is going to be somewhere down here. And so if it's, this many centimeters, I'm assuming this is centimeters here, then you just look for that point on your line here. So you draw a best fit line through your dots, and then you can say, and usually this is graphed versus the log of the molecular weight. Then you can say, okay, well then my protein must have a molecular weight that's somewhere in this region. Now, this is not a way to get the exact molecular weight, but you can get pretty close. And so again, you can figure out from the molecular weight, you can often figure out what kind of protein it is, whether it's the protein you're actually interested in or not. If you know the molecular weight of your protein, then you can say, well, if it migrated to the same distance that my protein would have migrated to, then that probably is my protein. On the other hand, if you don't know the molecular weight of your protein, this is a relatively easy way to estimate it. Now, again, SDS gels get rid of any charge information from a protein, so you have no idea, nothing about its charge is going to be present. But you can separate proteins by charge by doing what's called isoelectric focusing. And it's a little bit trickier. So obviously you don't want to use SDS or any other kind of denaturing molecule, because you want to separate the proteins by their pi. So, or the isoelectric point. So individual amino acids have isoelectric points, and individual proteins also have their own isoelectric point. Remember, that's the pH at which the net charge on that protein is 0. And so it's going to be a combination of the how many different charged amino acids are on there and sort of how they're positioned. But each protein should have its own kind of relatively unique PI. So to separate by PI, the first thing you have to do is to create a gel, and then in this case it's usually done with a gel that's a long thin strip instead of a big giant sheet. And you put an immobilized pH gradient onto that strip. So basically you set it up in a situation where one end of the gel has a higher pH than the other. So you then so one end, here in this case we've got a pH of 9 at one end and a pH of 3 at the other. And so you add your protein to one end, usually the end with the higher pH. At very high pH, most proteins will be negatively charged because they will have lost their protons, if they have any, to lose. And then you apply a current so that your protein starts moving through that gel. But the only thing that causes it to move, because again, there's no SDS here, the only thing that will cause the protein to move is those sort of inherent charges on there. And as it gets to lower and lower pHs, the protons will get added to those proteins, which means they will lose those negative charges until eventually they get to a point where their net charge is 0. And again, that will be that will correspond to the PI for that protein. And so the protein will stop. So what you'll have then is a bunch of dots along this gel that correspond to proteins that stopped when they got to their PI. So isoelectric focusing is just a way to separate proteins by their PI. And then you can combine isoelectric focusing and regular gel electrophoresis, or SDS gel electrophoresis. So after you do isoelectric focusing, then you can take that gel strip that has the protein separated by PI and put it on the top of a SDS gel, and then apply the current. So then the proteins, you'll have your proteins separated by PI along this strip, and then after you apply a current the other way, the proteins will move this way, and they will move basically at 90 degrees to the direction that they moved in the isoelectric focusing gel. And so we call this 2D gel electrophoresis because you're moving the proteins in two different dimensions. And the first dimension is PI, so again you separate by PI first, and then the second dimension, kind of the vertical dimension along here, will be molecular weight. So you have essentially a grid where if you go down the gel, molecular weight is decreasing, and if you go from left to right, PI is decreasing. So if you know the molecular weight and the PI of your gel, you can find it somewhere, of your protein, you can find it somewhere on that sheet. Another way to identify some proteins is by what's called specific activity. Now this only works for certain types of proteins. The protein has to participate in some sort of reaction. So this is commonly done for enzymes, but you can do it for any protein that participates in a reaction that you can measure. So a lot of enzymes, when they catalyze their reactions, they produce some sort of product that can be detected either directly or indirectly. Sometimes you can use special substrates for enzymes that turn colors when they react, either changing from one color to another, or their color goes away, or a color appears. And so you can measure the activity of that enzyme by how much color appears in your wells. There are other ways, other things you can do, other indirect ways to view other types of proteins as well. But each of these little wells here contains presumably an enzyme plus some sort of substrate that turns yellow when the enzyme is present. And so the intensity of the yellow color tells you how much of that protein is there. And when we say specific activity, what we really mean is the activity of that protein as a fraction of the total protein in a mixture. So the difference between activity and specific activity is like the difference between the red marbles in this beaker on the left. So this beaker contains red marbles plus a bunch of other marbles. This beaker does too, but the total number of marbles is much smaller, and the number of red marbles as a fraction of all the other marbles have gone up. And so over here you can see kind of a typical fractionation or purification process. So you start with your crude cellular extract, and you can find out whether there's 10,000 milligrams of protein total in that initial extract. And then if I measure the activity at that point, if I see 100,000 activity units, which is just a way of measuring how much the protein is active, then you would say your specific activity is 10 units per milligram. So then you do your various purification steps, you precipitate with salt, you do various chromatography techniques. What will happen is the fraction volume will go down, so the total volume you're left with will shrink because you're purifying, you're getting rid of stuff you don't want, and also the total amount of protein will go down. So you'll see less and less protein in milligrams total in your sample, because again, that's good. You want to see the total protein go down because you're getting rid of protein that's not supposed to be there. And the activity will go down too. So you're seeing less and less activity because none of these purification steps is perfect. Each one will get rid of some of the protein that you're actually after. But what should happen is your specific activity, if you're good at purifying, will go up. So our specific activity is getting larger and larger because the protein we actually want as a fraction of the proteins we're getting rid of will get bigger. And in fact, if you do subsequent purification steps and your specific activity does not increase, that tells you that you've probably purified that protein as much as you possibly can. All right, we will stop there, and next time we will talk about primary structure.